Okay, which brings us to today's main topic. We're going to be talking about immunity a couple of times here. We're going to be thinking about our immune systems and what we can expect to see there. I'm going to start with an example for you. It's a story about the boy in the bubble, which is a really interesting case of both understanding our immune system and understanding our medical and ethical framework around understanding the immune system. Then we're going to start breaking down the parts of the immune system. We'll go into the key parts of the innate immune system, inflammation, phagocytosis, and complement. And then we'll go into key parts of the adaptive immune system, antibodies that might see complex and cell-mediated killing. So keep in mind that immunology gets pretty complicated pretty fast. We're going to be kind of cruising across the scope of immunology pretty quickly here. So I want to make sure you guys are familiar with some of the most important things, but keep in mind there's always going to be more depth when it comes to a topic like this. Okay, so this is a picture of the boy in the bubble, and this is a pretty sad story. This is David Vetter, and what you can see in this picture is it's just a little kid, and he's sitting there in his diaper. He's got some toys, and then he's surrounded by this chamber. So, um, what happened here? This kid that was born in 1971, and he was born with severe combined immunodeficiency syndrome, or SCIDS. So this is actually, it's pretty rare, but it's even amongst its rarity, it's not always exactly the same. There are several different things that get classified in this severe combined immunodeficiency syndrome group. So there's multiple different causes that could come up with a similar set of symptoms. Basically, it means that his immune system was um, noted to be very compromised from the very beginning. So in order to protect him from the fact that pretty much any um, foreign microbe, even the harmless ones, would make him sick, they put him pretty quickly into this chamber setup. Um, Anything inside the chamber was sterile, so it meant that there was no living microbes whatsoever to come in contact with him. So water that he drank was sterilized, air was sterilized, food sterilized, diapers, toys, all sterilized before entering the chamber with him. Um, and the idea at the time of putting him into this bubble was that we would, as a medical community, come up with the answer to help him. And the unfortunate part to this story is that as people tried really, really hard to come up with something to protect his immune system, something that would allow him to come outside of the bubble, um, nothing worked. There was nothing that they could find in the years that he was in there that um, that would relieve this disease. So they were kind of running out of options, and around age 10, 11, they started to realize that they couldn't just allow this kid to live his entire life inside the bubble. So they went with something that had the possibility of working, but they had already previously known wasn't necessarily the best case scenario. We talked before about where the generation of white blood cells comes from in the body, and it comes from bone marrow. So the idea was that maybe they could um, provide him with a competent immune system if they just transplanted stem cells from the bone marrow of a donor. The closest donor that they could find was his sister in terms of matching um, genetic type. And unfortunately, they gave the bone marrow transplant to him from his sister, and he died 2 weeks later. Um, Inside of her bone marrow was a dormant virus called the Epstein-Barr virus. Some of you may have heard of this. It is the cause of a disease called mono. So if you've ever heard of anyone getting mono, that's from transmission of Epstein-Barr. Mono is sometimes known as kind of like the kissing disease. So, it's fairly common amongst teenagers or preteens. So this was his older sister. It means she had contracted mono at some point. She may not have even known that she had it. So it was sitting there in her cells, even in her stem cells, sitting dormant. And so when they transferred things over, they were transferring a pathogen as well as the bone marrow. Okay, so give you guys that example, and it's a very dramatic example for sure, to try to show how complex the immune system is, and how we still definitely do not understand it completely. We can understand some parts, but there's a lot that still is mysterious to us. The immune system has a really tough job. So, one, it's trying to protect you against any exposure to this growing list of viruses, bacteria, fungi, worms, and other eukaryotes. This is the numbers here are saying like the known pathogens that we have. We know there's over 2,000 viruses that the immune system has to be able to come up with a response to, or the viruses can be lethal. Same thing with bacteria and on down the list. And in each of these cases, you don't get the defense from your parents. You have to come up with a defense on your own immune system protecting you. That's not all the immune system does. It also is protecting you on pretty much a daily basis from cancers. So um, this is hard to realize. It's another one of those invisible things that's happening, but pretty much all of us at some point will have a cell that goes to the route of cancer, where it's going to start dividing and could potentially become a tumor. The immune system actually catches a lot of these and is able to induce senescence or kill a cell that is otherwise going to go on to be a tumor. So we have our immune system protecting us against cancer. And of course, when someone is actually diagnosed with cancer, it means that it's a case when the immune system has missed it or been unable to stop that cancer. It also helps in the removing of damaged tissue. So anytime you're healing, the immune system is going to be getting involved to take care of whatever's happening at those sites. And the other thing to keep in mind with the immune system is that uh, even as it's preventing harm, the immune system can also cause harm or cause disease. Examples of this are autoimmune disease, anaphylaxis, and problems with transplants. So I've already mentioned a couple times now, anytime that the immune system is overactive, it can lead to problems. Okay, so I want to give you guys some of the players to know in the immune system. We're going to take these at a couple of different levels in terms of response. So we're dividing up the immune system based on like what is kind of hanging out all the time, what makes the first response to activate the immune system, second response, and then later responses. So we're going to start off with sentinel cells that just hang out in your tissues. Then we'll talk about circulating phagocytes and lymphocytes. The sentinel cells in your tissues are things like dendritic cells, macrophages, and mast cells. Their job is to provide kind of a 24/7 round the clock protection and make calls for help. So dendritic cells, macrophages, and mast cells, these are all just sort of like dispersed throughout, especially your skin tissues, although they can exist in other places too, but kind of hanging out in the uh, basement layers of your skin are going to be cells trying to protect and watch out for anything that's happening. So dendritic cells play an important role in antigen processing. So they will help to come up with some potential antigens, and then they present it to other cells in your immune system. So they're like finding things that could be considered foreign and then sort of like passing the message along. Macrophages are doing kind of a basic grunt level work of trying to use phagocytosis to gobble things up. We'll cover phagocytosis in a second. So that's mostly targeting viruses and bacteria. And then mast cells, you may be familiar with if you have allergies. So mast cells are involved in say, histamine production. So if you're like me, I have a lot of allergies to like dust, mildew, things like that. And what's happening there is that my mast cells have detected something, and they'll release histamine and start to cause an immune response. This, of course, can be really frustrating because there's times when, if it's just like mildew, I don't really need an immune response to come up. And so then I take a drug called an antihistamine, and an antihistamine is just trying to block that histamine signal while the mast cells are trying to ramp my immune system up. An antihistamine will ramp your system back down. And again, if you're like me, put me to sleep as well. Okay, so these cells all have a job of protecting and then calling for help. Because they can't do like the full immune response on their own, they can just get things started. Then we're going to have our circulating phagocytes, neutrophils, monocytes, and eosinophils. Neutrophils are often first responders to an infection, like say if you get a cut. Neutrophils are really powerful cells. I'll show you an example. We'll watch a video of a neutrophil in a second, but neutrophils are really good at being generalized attackers of bacteria. They can also do phagocytosis as macrophages can, and uh, neutrophils are one of the things that you can actually kind of see them. One of the ways that they'll attack uh, bacteria or mount an immune response is they can do something called explosive cell lysis, where they basically kill themselves and in the process release a whole bunch of nasty things into their local environment. And sometimes, rather than doing phagocytosis and like gobbling up one cell, they'll just decide, okay, I'm surrounded by bacteria, so I'm just going to do this explosive cell lysis thing and hope to kill every bacterial cell around me. And it works quite well. And in the process, it leaves behind kind of an immune system form of wreckage, and you would recognize this as pus. So neutrophils exploding is what produces pus. So you can kind of see that yellowy, gooey, uh, very icky substance is uh, exploded neutrophils and the remains behind. A second responder is going to be the monocytes. They can actually do two things. They can either help out the neutrophils in clearing up an infection, or they can go and signal for more help. And then we've got eosinophils. Eosinophils are um, kind of dedicated to the difficulty of larger pathogens, so worms, eukaryotic parasites. Um, since those can't be tackled with phagocytosis, eosinophils make use of some alternative mechanisms to try to handle the larger stuff. Okay, and then finally we have the lymphocytes. And the lymphocytes are going to be a wide range of cells. I have 4 of the cell types down here. We've got our helper T cells, B cells, cytotoxic T cells, and natural killer cells. Hopefully you guys have heard these terms before. We're going to go into a little bit more detail on them a little bit later. So your helper T cells are basically helping to promote a good antigen response. The B cells are going to be responsible for producing serum antibodies. These are antibodies that are moving around freely in your bloodstream and other tissues. And then the cytotoxic T cells are going to be T cells that promote cell-mediated antibody-based killing. So they hold on to the antibodies in their cell membranes, directly make contact with the cell, and then destroy it. And then natural killer cells are able to do a variety of killing, of destroying many different cell types. That crosses over from the infectious diseases into, some of the things like stopping cancers. Let me pause for 1 second and just see if there's any questions out there from anybody. Okay, I'm going to assume you guys are following along and hopefully getting lots of notes down and digging in on these immune system terms. There's another way to divide up the immune system instead of based on who responds first, second, etc. You can break the immune system down into the innate and the adaptive immune system. That's also considered the non-specific or the specific response. You'll see this division of things many times. So on the innate immune system side, that's going to be covering everything from inflammation, phagocytosis, the complement, cytokines, macrophages, neutrophils, and natural killer cells. And then on the adaptive immune system side, we're going to have the major histocompatibility complex, the lymphocytes, um, T cells, B cells, and then antibody production. And the thing is, this designation is um, a little bit of us just trying to put things into groups. Like, it all works together simultaneously, and there's a lot of communication between these two areas. And there's things that are kind of involved in the innate side, but mostly involved in adaptive, or mostly involved in innate, but sort of involved in adaptive. There's lots of crossover back and forth too. But basically what we're thinking about here is that there's a general response that can work against literally any foreign entity. And that's really useful to have as like a very like quick response, or just sort of all the time response. And then the adaptive side is what um, allows us to have a memory of things, and to be able to prevent getting sick from something twice, and to be able to really, like, for any very, very tricky pathogen or something that's really causing an overwhelming response in the body, to have a very focused and dedicated response that can be ramped up very high, and then go after that one specific pathogen. Okay. And this is what that whole vaccine technology is based on, is being able to get an adaptive response so that you can hold on to the memory. Okay, so I'm going to talk about a couple of the innate responses first. One of them is going to be inflammation. So it starts with our dendritic cells again. So our dendritic cells are out there trying to detect anything that's generally foreign. So basically, they're on alert, and they don't really know what they're looking for. They're just looking for anything that just doesn't look familiar to them. They're going to be one of the cells that can secrete cytokines. Remember, that's the term that we saw with the 1918 Spanish flu, of the cytokine storm. Cytokines are proteins that are cell action proteins. If you look at the roots of the word, basically they're trying to get other cells into action. So they send a message, and they promote the ramping up of the rest of the immune system. Then we're going to have our blood vessel cells get involved, and they're going to secrete something called adhesins. And adhesins are proteins that are sticky. So sticky protein allows things to adhere. So when your blood vessels are secreting adhesins, they're going to be trying to get some of those circulating phagocytes and lymphocytes to stick to one particular site instead of bouncing all around the circulatory system. You want them to just like hone in on one area. So that's going to allow your immune cells to adhere to that one particular site and then start defending. And, overall, inflammation is generally good. It is a very useful part of our immune response. But inflammation is something that can go too far, and is something that can on its own cause damage when it's overactive. So a lot of um, our understanding of inflammation is based around trying to moderate our inflammation response to get it to be at just the right level. Okay, so I'm going to show some specifics here, okay, including someone with really bad inflammation occurring in the eyelid, and then a diagram of our inflammation mechanism. We're going to focus on that part. So as we go into our inflammation mechanism here, what you're looking at is kind of a cross section of blood vessel all the way through to skin, to the outer layer. So the blood vessel on the left is trying to show that there's going to be some red blood cells hanging out in the circulatory system, and there's going to be some larger white blood cells there. And then there's this dividing point, or this the barrier of the blood vessels themselves, and then all the rest of the tissue of the skin. So what we're looking at here is like, okay, maybe it's just like a little paper cut. So, little cuts like that can happen at any time, and we don't want it to be lethal. So we're going to mount an immune response. The immune response is going to start with in this case, mast cells, signaling with histamine to recruit other cells. So those two purple mast cells there are going to send out a burst of histamine, that some of which will get into the blood vessels. This is going to help recruit cells, and it's also going to increase the blood vessel permeability. This is not what you normally want. You normally want your blood vessels to stay as like a solid layer, but it's an amazing and important response that you can actually sort of remove the strictness of the barrier and allow some of the blood to leak into this area of the skin. Of course, this would make the skin start to look sort of red and swollen from that um, action. You can see the pathogens in green there in this picture. And so basically what the immune response is going to try to do is to allow some of the white blood cells, circulating phagocytes from the circulatory system to cross that blood vessel barrier, make contact with these pathogens, and start finding ways to either phagocytose or kill them in other ways. So then we're going to have our phagocytes are going to bind to adhesins, they're going to migrate into the wound. This is where we might get some of those neutrophils showing up to cause explosive cell lysis and form some pus. Okay, so that's kind of like the bigger picture of what's happening. And if we go into the smaller picture of things, we'd be looking at how cell types like our macrophages would try to identify and engulf foreign cells. And again, this can happen as part of innate immunity. It doesn't need any specific signal. The way that this works was discovered by this scientist, Mechnikov, and you can see the diagrams down below. What you'll have is a pathogen interacting with phagocyte surface receptors on the outside of the macrophage. And these surface receptors are like, kind of all over the cell. And as soon as they make a solid contact with something they recognize as foreign, they'll um, they'll sort of rearrange the actin cytoskeleton around that cell. They'll be able to engulf that cell, bring it into a phagosome. The phagosome is going to merge with lysosome, which is going to have all sorts of nasty molecules in it. And the hope is that inside that phagolysosome, uh, anything inside of it would die, and then sort of like chop up the pieces and kick the pieces back out. Okay, so for phagocytosis to work, we're going to have to recognize, ingest, and then kill. Um, it's going to use everything from surface receptors, cytoskeleton to power ingestion, killing with digestive enzymes from lysosome, and then this final thing that I hadn't mentioned yet, which is opsonization. Um, we can use antibodies, which is part of the adaptive immune system, and proteins from the complement to coat a microbe to make it easier to promote phagocytosis. So that's part of the way that those surface receptors are triggered to do the process. All right, now I'm going to try to show this with a short movie. Okay, so this movie is short, but it's pretty fantastic. So we'll watch this a couple times. What you're looking at here is a tissue culture, and someone noticed that in their culture of um, blood cells that they had this one neutrophil that they could track. So the large spherical objects or sometimes spiky looking objects, those are red blood cells. We'll show this again. And then the neutrophil is the very large amorphous moving thing. Now you can see that the neutrophil is chasing after something. That's a Diplococcus bacterial cell. So it gives you a sense of the size differential between typical bacteria and our typical blood cells. The bacteria are very small in comparison to the blood cells. And what's happening here is we get to see the chase. I'm going to show it one more time as soon as this repeats. Oop, there we go. Okay, so if I pause right here, this is our Diplococcus bacterial cell. These are red blood cells that cannot move. So they're just kind of like obstacles in the way. And then we have our neutrophil, which can rearrange its cytoskeleton in order to move and chase after a bacterium. You'll notice a couple of things happen here. One, There's moments like this where there's actually a Diplococcus bacteria up above the neutrophil and another one down to the lower left. And you'll see the neutrophil has this moment of indecision where it actually tries to move towards both. And fortunately, it knows that it cannot chase two birds at once. So it'll hit a point where it makes up its mind and it says, well, I'm just going to chase after this one. I'm going to have to deal with the other one later. So you'll see it sort of decide, okay, I can only go after one. So then after going after just the one, it's actually following a chemical trail where the bacteria is releasing just enough little molecules that look like bacteria that sort of give the neutrophil the scent for it to chase after. And then you see when it finally makes contact, the bacteria stops moving, and you will see the very beginning part of the phagocytosis process as the cells get engulfed and are now inside of the neutrophil. And again, that's what you would picture is happening. In this case, the neutrophil can move pretty well because the environment's pretty loose and unencumbered. But neutrophils will try to move through your skin cells in a similar way to try to chase down any little bacterium that they can find. Okay, there's the link so you guys can watch that video again anytime. Okay, so I've been talking nonstop about the neutrophils, and remember, these guys are considered first responders. They're great at killing microbes. They produce lots of nasty chemicals, but they also damage tissues and themselves because they're so powerful or so toxic. So you don't want to have neutrophils active all the time. You really only want to recruit them when there's something foreign around. Our second responders are the monocytes, which I mentioned before. And remember, their job is going to either be to continue the fight against whatever the invader is, or if they detect that everything is kind of taken care of, then they can switch to a cleanup mode. So they have a little bit more flexibility than the neutrophils do. They can either do the same thing or they can start a cleanup. Okay, I've mentioned this a couple times now. The complement system is really fairly complicated, but I want you to make sure you guys know just a little tiny bit about it. It's the heat labile component of the innate immune system. Heat labile meaning that if you heat it up, it stops working. So that's because it's based on proteins, and when proteins get heated up, they misfold, they denature, so you can inactivate the complement system through heating things up. There's going to be lots of different pathways happening, but I'll give you guys one example here. The cleavage of the C3 protein is an important part in activating the complement system. It's a positive feedback loop that increases complement activity. The C3 protein will divide into two chunks, C3b and C3a, so it's two smaller proteins after some protease activity. The C3b part of the protein helps with opsonization. Again, that's coating foreign bacteria so that the immune system can phagocytose them and triggering of pore formation in blood vessels. And then the C3a component is involved in T cell activation and inflammation control. Okay, so it's just one example of how there's proteins hanging out, and the C3 protein, when it's in its large form, isn't really signaling anything. But when it gets clipped into those two smaller forms, they'll immediately go off and signal these two different pathways. So you have the ability to have a signal, a messenger that's kind of around and available all the time in an inert form, and then you crack it open, and then it goes off and does these other functions. Okay, so I'm going to leave that. I'm not going to go into any more of the complexity of the innate immune system. I'll just leave it there with inflammation, phagocytosis, and complement. And then I want to give you guys some info on the adaptive immune system. So this is where we're going to have specific immune responses. We're going to involve our MHC, our major histocompatibility complex. We're going to involve our lymphocytes, and then we're going to get into antibodies as all part of the innate, or as part of the adaptive immune system. We've only got 10 minutes left, so we're not going to cover all of this today. But again, this is where we're thinking about how does our immune system get to be specific, And how does it get to have memory? Okay, so I already gave you guys this term antigen. That's an antibody generator. So what we're going to have to do is we're going to have an antigen, and here it's the little light blue star shape in the picture, and it's going to have to find a couple of different cell types. One of them is the B cells. So our B cells can produce antibodies that will be free in the bloodstream, or um, this is sometimes called the humoral immunity, the antibody part uh, of the blood serum. So antibodies bind to antigens. And now, in order to have the right antibody for antigens, we're going to go through these different steps. We're going to have a production step, we're going to have filtering, we're going to have selection and storage. In the end, we're going to end up with memory B cells. And memory B cells are what give your immune system the ability to have rapid antibody production, or in other words, when we talk about someone having immunity to a virus or to a disease, the immunity comes from those memory B cells that they've already seen an antigen before, and so they can very quickly respond to it again. The amazing thing here is that this can be years later. So you can be exposed to something like chicken pox as a kid, and for years afterwards, your immune system will remember the antigens of chicken pox. And so, you can be an adult hanging out with a sick child that has chicken pox, and you'll be exposed to the virus again. And hopefully, your B cells, your memory B cells, are there hanging out and ready to respond to it. So you don't even develop any symptoms because you respond to it so fast. Okay, so what we're going to be looking at here is kind of thinking about where things are happening and what needs to happen. And the very beginning, the production part, is going to be happening in the bone marrow. That's where we're going to be going from stem cells to immature B cells. And then after we uh, filter our B cells out, so we release them to where we're going to allow them to be part of our immune system, you'll have these repositories in lymph nodes and in the spleen where B cells will be kind of hanging out and ready for detecting of antigens. And these B cells will be present in these populations where there's actually lots of different B cells, and each of them has on its surface a different antibody. And so the picture here on the left is trying to show that, where you've got, in this case, it's just two B cells, one that can bind to the blue star shaped antigen and one that can't bind to it. In reality, there would be thousands of different B cells. So you'd be looking at trying to display this antigen to these thousands of different B cells, hoping to find one that makes a strong connection. Okay, so let's say we found one that makes a strong connection. It's holding on to that antigen now. We want to give it further encouragement to say, like, yes, you're the one. This is the B cell that we want. This is the antibody that we want. So we're going to make use of helper T cells. And you can see the helper T cell is there to say, like, yep, this looks like a good connection. I'm going to promote this B cell to grow and produce more antibody. And then you'll see on the far right, the B cell, as it grows, gets this massive size and becomes a plasma cell. And plasma cells are antibody factories. So these plasma cells build lots and lots and lots of these antibodies. You can see them releasing the antibodies, and then those antibodies can go around through the bloodstream. And then the hope is that they'll continue binding up to that antigen signal anywhere they go and help to promote the immune response wherever they find that signal. Okay, I want to show you guys a little bit more of the detail on antibody structure. So antibodies can also be called immunoglobulins, and then immunoglobulins can be abbreviated as IG. And then there are several different um, immunoglobulins. So there'll be like IgA and IgB and IgG. So IgG is the most common or the most numerous immunoglobulin that you'll have in your bloodstream. It consists of two light chain proteins and two heavy chain proteins. So it's four separate polypeptides all coming together into one functional unit. Overall, it has this kind of like really thick Y structure to it. And in this Y structure are going to be some really important segments. There's going to be a variable region and there's going to be a constant region. The variable region is at the top in this picture, and that's the part that is binding to antigen. Okay, so you can see they've got little antigen binding site arrows there. Then down below, we have our constant region. Okay, so that part is definitely not going to be binding to antigen. And the key thing here is that the antibody is providing a bridge. It's trying to connect your immune cells to an antigen or to a pathogen. To do that, it has the top part binding to antigen, part of your pathogen, and the bottom part, the constant region, binds to immune cells. So it doesn't say on this slide here that it's binding to immune cells, but that constant part, it means that our immune cells can recognize every antibody as having that same constant region, and it gives them the signal that they need to know that they've like found an antibody. Okay, you can also look at this picture to try to go back and forth between the sorts of diagrams and drawings that people typically do to try to, represent an antibody, as well as the protein ribbon structure on the right to show what an antibody really looks like or is a little bit closer to what it really looks like. And so again, what I want you guys to get from there is that you can kind of see that the antigen binding sites or the variable region have these like flexible domains that allows them to sort of like move around, and different bacteria or viruses will have different shapes and it can help to bind to those different shapes, and then it stays connected to that constant region down below, which is going to be really critical for recognition by your immune system. Okay, like I said, there's several different antibody types. We've got IgG, IgE, IgD, IgM, and IgA. IgG is our most common, and so that's the structure for antibodies you will see the most often. You can see that IgE and IgD basically have the same structure of that like kind of thick Y shape. And then you can see that IgA and IgM, they're very different. They have a different structure. IgA is kind of two Y-shaped structures stuck together. And then IgM is actually a pentameric structure. It's got five of those Y-shaped structures all together. And those different structures help to accomplish different jobs, with IgM being the most different job, where IgM is really good at helping to target clusters of bacteria. And so you can kind of see from that pentameric structure that it's going to be able to grab onto lots of different things. IgA also is kind of an intermediate between IgG and IgM. Okay, so I'm going to give you guys a little bit of a breakdown on these immunoglobulins, and then we'll wrap up here. So IgG is 80% of your total serum immunoglobulin. That's why it's the one we focus on the most. IgM's pentameric structure actually helps it to be part of the first responder class. So we make a lot of IgM to just sort of help to get the immune response started. IgA is really important in sort of difficult to reach areas. So it hangs out in mucus, saliva, tears, and in milk. And so IgA is providing a protection in areas like outside of the bloodstream. IgE seems to mostly be involved in helping to interact with our sentinel cells and allergy responses. And IgD is part of B cells and helps them in their ability to promote formation of IgG. Okay, guys, so I'm going to post slides up to here in this question, and the question I'll let you guys think about for next time is what's the difference between when our immune system responds to a bacterial pathogen versus a viral pathogen.